The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
So what we're going to do now is look at the summary of what we just talked about. So in the first step, or in the first stage of this reaction right here, we have glucose actually getting converted into glucose 6-phosphate. Now this reaction requires the input of energy in the form of ATP. And what happens is that this reaction is catalyzed by the enzyme hexokinase. So that's the first step right there. Now in the second step, glucose 6-phosphate is converted into its isomer, fructose 6-phosphate, and the enzyme responsible for that step is phosphoglucoisomerase. Now in the next step, which is the third step, fructose 6-phosphate is converted into fructose 1,6-biphosphate. This third step requires the input of another ATP molecule, and the enzyme responsible for catalyzing that step is phosphofructokinase. Now after that, fructose 1,6-biphosphate in the fourth step is broken down or cleaved into two molecules, one glyceraldehyde 3-phosphate and two dihydroxyacetone phosphate. Now in the fifth step, the dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate under the activity of the enzyme triose phosphate isomerase. Now why this is important is because the only molecule that can proceed into the next step of the reaction is glyceraldehyde 3-phosphate, and for that reason the dihydroxyacetone phosphate that is produced must be converted into glyceraldehyde 3-phosphate. Okay, so step 4 converts it into those two molecules right there, and step 5 converts the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So at this point in time, going into step 6 right here, we have two molecules of glyceraldehyde 3-phosphate. So anything that happens from this point forward is doubled. So in this case, we have our glyceraldehyde 3-phosphate molecules, each of them being converted into two molecules of 1,3-biphosphoglycerate, and the enzyme responsible for catalyzing that process is glyceraldehyde phosphate dehydrogenase. Now, after our two molecules of 1,3-biphosphoglycerate have been formed, another enzyme, which is phosphoglycerate kinase, will convert 1,3-biphosphoglycerate to 3-phosphoglycerate. Now, this process actually results in the generation of energy in the form of ATP. And as you can see, at this point in time, our first two ATP molecules are synthesized or produced from each of our glyceraldehyde 3-phosphate molecules. Now, that is basically step 7 that I just talked about. Now, in step 8, our 3-phosphoglycerate formed in step 7 is then converted into 2-phosphoglycerate. So it's basically a migration reaction, and the enzyme responsible for this is phosphoglycerate mutase. It just shifts the position of that phosphate group from the third carbon right there to the second one, resulting in the formation of 2-phosphoglycerate. And in the 9th step of the reaction, 2-phosphoglycerate is then converted into phosphoenol pyruvate. It's converted into phosphoenol pyruvate, and the reaction the enzyme that is responsible for this reaction is referred to as enolase right there. Right? That's step 9. And in the last and final step of this process, which is step 10, our phosphoenol pyruvate is then converted into pyruvate, and the enzyme is pyruvate kinase, which catalyzes the transfer of a high energy phosphate group from phosphoenol pyruvate to ADP to form ATP, which results in the formation of our pyruvate molecule. So two of these from each glucose molecule. So remember, after we form those two glyceraldehyde 3-phosphate, everything that occurs from step 6 onwards through to step 10 happens in twos. So that's a very important thing to remember, because that's how we get our net gain of 2 ATP, because we use 2 and we generate 2. We also produce 2 NADH in the process, and that's from here. So each one of those glyceraldehyde 3-phosphate conversions into 1,3-biphosphoglycerate will produce one molecule of NADH. That happens twice, so we have 2 NADH, and we also have 2 pyruvate produced from that entire process. So I hope glycolysis, in a nutshell, is something that's more palatable for you right now. If there's anything you didn't understand, or if I was moving a little too quickly for your liking, just go back. That's why you have this lecture right here. That's why you have that table of contents. You can just go back to anything that was a challenge and review it.